Good morning, students. Today in this video we will be discussing about the reproduction in Riccia. In case of Riccia, it reproduces by vegetative means as well as by sexual means. First of all, we will discuss about the vegetative modes of reproduction. Vegetative reproduction in case of Riccia takes place by several methods like fragmentation, formation of adventitious branches, persistent growing apices, formation of tubers by the death of the thallus during drought or by the rhizoids. So, these much of different modes of vegetative reproduction exist in case of Riccia. Fragmentation is familiar to you all. That is, the main thallus, it divides into different fragments, and each bit of fragments it will be converted to an individual thallus or individual plant. That is fragmentation. And next is the formation of adventitious branches. That is, in case of Riccia fluitans or several other species of Riccia, adventitious branches may develop on the main thallus, and these branches, if they are separated from the mother plant, they can grow to a new plant. That is known as vegetative mode of reproduction by means of adventitious branches. And another type of vegetative reproduction is by the death of the thallus during drought. That is, when a prevailing drought condition exists, then what happens is the main thallus or the mother plant, all parts of the thallus except the growing tip, that is the apical portion, except that apical portion, all other parts of the thallus die out, and the apical part, which is still alive, it grows deep in the soil and becomes thick. It becomes active and grow during the next growing season and develop to a new thallus. This process is actually a method of perenniation. It is trying to cope up with the unfavorable condition. It grows to a new thallus when the upcoming favorable season comes. So this is another mode, and next is by the tubers. Certain species of Riccia, in them the apices of the thallus, it forms a small structure like a tuber, and these are the dormant structures or the dormant tubers which remain dormant when the unfavorable period comes, and again regain the growth when the favorable conditions arise. So that is another mode of reproduction by means of tubers. And the next one is by rhizoids. In some cases, the apical region of the rhizoids, they divide and form a cellular mass. They repeatedly divide and they form a mass of cells. And these cells possess chloroplast and have the capacity to grow into a new thalli. That is another form of vegetative reproduction. So these much of different varieties of vegetative reproduction exist in case of Riccia. And the next type is sexual reproduction. Sexual reproduction in case of Riccia is oogamous. It takes place by means of a male and female reproductive organs. The male reproductive organ is known as Anthridium, and the female organ is called as Archaegonium. Most of the Riccia species, they are homothallous and with the male and female organs on the same thallus. That is, we can call it as monoecious. Okay, some species are there which are dioecious. And Let's see the development of Anthridium. Anthridium is the male reproductive organ, and it is a club shaped in structure. It attaches to the bottom of the anthridial chamber by a short stalk, and the anthridial chamber, it opens to the outside by a narrow pore. This narrow pore, that is known as ostiole. The body of the anthridium, it consists of a central mass of fertile cells called androcytes, or we can simply call it as anthrozoid mother cells. They are covered by a one cell thick sterile, you can see it here, one cell thick sterile layer. That sterile layer is called as the jacket layer. Each of these endocytes, they become an anthrozoid. During the rainy season, this jacket layer, this one cell thick jacket layer, it imbibes water and they get dissolved and releases the anthrocytes or the anthrozoids to the outside. And these anthrozoids, they swim in the water, and they go in search of the Archaegonium. So this is all about the anthrozoids or the structure of Anthridium. So Anthridium is a club-shaped structure. It is found inside an anthridial chamber. It is attached to the base of the anthridial chamber by means of a short stalk, and it is covered by a single unicellular uniseriate one cell thick jacket layer, and inside these are the androcytes or anthrozoids, which are formed from the anthridial mother cells. And upon the upcoming rainy season, what happens is the jacket layer, it imbibes water and dissolves and releases the anthrozoids to the outside, and they swim in the water and go in search of Archaegonium. And next is the this is one androcyte and anthridium mother cell divided into two androcytes. So one cell, it divides to two cells to form two anthrozoids. Anthrozoid is a spiral structure. And next is the formation or the structure details of Archaegonium. Archaegonium, it is a flask shaped structure. So it is a flask shaped structure. Here also the Archaegonium is attached in the similar way like that of the Anthridium. That is, it is attached to the bottom of the chamber by a short stalk, and it has a basal swollen region, that is the venter, and the upper elongated and narrow neck region is there. So it has two regions, a swollen venter and a elongated neck region. The neck is composed of 6 vertical rows of cells. The neck region, it is formed of 6 vertical rows of cells, and within the neck is the uniseriate row of 4 to 6 neck canal cells. Okay, so within the neck region there are 4 to 6 rows of neck canal cells. The tip of the neck is covered by 4 cells. They are known as cover cells. The tip of the neck region, it is covered by, this is not the actual Archaegonium. Actually the upper part is not visible in this figure. So the upper region of the neck, they are at the tip of the neck there will be 4 cells, that is known as the cover cells. Here also the venter is covered by a one cell thick layer, sterile layer, that is known as the jacket layer, and it encloses a large and naked egg. It encloses a large egg inside it. And also with the egg we can find one more cell, that is the venter canal cell. So the cells found inside the neck region are called the neck canal cell, and one with the egg is called the venter canal cell. The upper part of the Archaegonium, that is the neck region, it is actually it protrudes out, or it protrudes away from the venter. And now coming to the fertilization process, I have already mentioned that the anthrozooids which are released from the Anthridium, they are released to the water and they swim through the water in search of Archaegonium. And as the Anthridium as the anthrozooids, they approach or they come in the vicinity of Archaegonium, the neck canal, at that by the time the Archaegonium will be mature. And what happens during the maturation, the neck canal cells, these canals, the canal region or the cavity is filled by the neck canal cells and the venter canal cells, right? So the at the time of maturity, the neck canal cells and the venter canal cell of the Archaegonium, it disintegrate and form a mucilaginous substance, and this mucilaginous substance is the one which fills this canal region. And this mucilaginous substance, it imbibes water. This force, this imbibitional force within the neck, it causes certain kinds of separation of the cover cells from one another. And so the tip region, which is occupied by the cover cells, that will be separated from each other, forming a narrow passage to the inside through which the anthrozooids can enter. So from this passage, the anthrozooids can freely enter into the Archaegonium and they can come in contact with the egg that is found inside the venter. So the anthrozooids enter the neck and only one of them fuses with the egg to form the zygote, which is the diploid form. So the zygote is the first cell which represents the sporophyte. The diploid zygote, it is the mother cell of the sporophytic generation. What happens is this zygote, it secretes a wall around it and increases in the size. It occupies the whole cavity of the Archaegonium. It divides anticlinally and periclinally and form a two-layered calyptra. See, this is the calyptra. So it enlarges in size to occupy the whole venter cavity, and it divides anticlinally and periclinally to form a two-layered calyptra around the developing sporophyte. So the mature sporophyte is represented by the spore sac, and it lacks both the foot and seta. Spore mother cells are enclosed by a single layered jacket, the capsule wall. And the capsule, it is the protective covering of calyptra, which is a part of the gametophyte. The first division of the zygote is transverse. First division is transverse. See here, it is transverse. So the first division is transverse, and it may form two more or less equisized cells. So first of all, it divides transverse, then it divides another division which is vertical and forms a quadrant stage or the quadrant embryo. Later it forms octant stage, and several other subsequent divisions also occur and form a 20 to 40 cells in number. And the superficial cells in this mass, they divide periclinally to form the outer amphithecium and the inner endothecium. So the endothecium is repeatedly dividing, and it forms the sporogenous mass of cells. And from these sporogenous mass, each of the functional spore mother cell, it undergoes reduction division to form 4 haploid cells. And these spores are not liberated immediately after formation, and instead they are set free after an year, only after 1 year or so. And the spores are liberated by gradual decay of the capsule wall. Gradual decay of the capsule wall, and the capsule wall, it disintegrates before the division of the spore mother cell, and after some time the inner layer of the calyptra, it also degenerates. So there is no particular mechanism for dehiscing these kinds of spores. The spores are not liberated immediately after formation, but they remain in the spore sac till it gets dead and decayed, and only after the death and decay of the spore sac it is liberated out, and it is carried by the raindrops, splashing raindrops, and when the favorable condition arrives they germinate. Okay, so regarding the germination of spores, these spores, when they germinate only when there is adequate water, adequate amount of water around in the surroundings, and the exosporium and mesosporium, it ruptures the spore. It has certain walls. Its walls is differentiated into 3 layers, namely exosporium, the outer one, mesosporium, the middle one, and endosporium, the inner one. So during germination what happens, it absorbs water, and the exosporium and mesosporium both ruptures, and the endosporium, the inner wall endosporium, it comes out in the form of a tube. You can see here it comes out in the form of a tube, and this is called as germ tube. The germ tube, it is filled with abundant amount of cytoplasm and certain oil globules and chloroplast also. It elongates rapidly and forms a club shaped structure, and a transverse wall at the distal end is formed, and a small to separate a small cell which undergoes 3 vertical divisions. Later on, several divisions occur to form 2 tiers of 4 cells each, and one of the tiers, it becomes apical cell, and it regularly it forms newer cells. Okay, so the germ tube with a thin filamentous rhizoid develops from the base of it, and this is the rhizoid. You can see here germinating forms the rhizoids. The upper region continuously divides and forms the sporophyte. So this is all about the reproduction in Riccia. Okay.